Hello everyone, I'm Islam and I'll be explaining the sensory receptors part 2 lecture. So just a heads up, I won't be explaining the experiments which the doctor adds to the slides, so you can just go through them on your own. So let's begin here. Type. We have the nerve fibers, and nerve fibers, they come in different sizes and they can either be unmyelinated or myelinated. And because of that, they conduct different sensations at different speeds. So you can see here, these small ones are the unmyelinated axons, and this large one is a myelinated axon surrounded by the myelin sheaths. And even the myelin sheaths themselves, they come in different thicknesses. So this one you can see is thicker than this one. And the thicker and larger the axon and myelin sheath is, the faster a certain sensation is conducted. So by now I'm sure you're familiar with this graph. And as you can see here, the sense of position is the fastest sensation to be conducted. Why is that? It is because your brain needs to always know your exact position to be able to send a precise motor signal for your next move, whether in walking or in running. So other sensation can arrive slightly later because your brain can afford to send a delayed response, but it cannot do that with the sense of position. So in the part 1 lecture, we talked about the pathways in which the sensations travel. We have the dorsal column system and the anterolateral system. The dorsal column in the gray is ipsilateral, meaning it travels on the same side which the stimulus is in the spinal cord. So if the stimulus is on the left, it travels in the spinal cord on the left. And in the anterolateral system, it is contralateral, and the signal travels to the opposite side and travels there in the spinal cord. So the size of the nerve fibers in the dorsal column system is larger, meaning the conduction velocity is faster. However, that's the opposite for the anterolateral system. So the size of the nerve fibers is smaller, making the conduction velocity slower. And these are the sensations for which travel in each pathway. I think they're clear over here. Yes. So we have the ones in the red circles travel in the anterolateral system, and the ones in the gray circle travel in the dorsal column system. And to avoid any confusion, because the other lecture, the part 1 lecture, said that pressure travels through the dorsal column system, so this is true for the light pressure, because light pressure travels through the dorsal column system. However, the deep pressure travels through the anterolateral system. And also for touch, refined touch travels through the dorsal column system, and the crude touch travels through the anterolateral system. And if you take a look at these sensations, you can see that most of them are thermoreceptors and nociceptors. And you can see that the gray ones are mechanoreceptors in the dorsal column system. So how are the other two receptors, the photoreceptors and chemoreceptors, sent to the brain? They don't actually take these two pathways, because as we know from the other lecture, the part 1 lecture, the photoreceptors are found in the eyes and the chemoreceptors are found in the mouth, meaning they are very close to the brain. So they don't need to travel through the spinal cord, and they can just directly go to the brain. So how do we test these pathways? For the anterolateral system, we can simply test the pain or pressure with a pinprick for pain, and having two warm and cold objects, and having the patient see if there is a change in the temperature. However, for the dorsal column system, we need to apply something called Romberg's test or Romberg's test. So to maintain our balance we need two of these three factors. Vision, or the vestibular system, which is the inner ear, and the proprioception, which is the sense of position. So you'd ask the patient to close their eyes. This way we eliminate the vision, and the patient would need to depend on these two for balance. So if the patient manages to maintain his balance, you'd know that these two are working just fine. However, if the patient sways and cannot maintain the balance, we know that there's something wrong with one of these two. And in the case of Romberg's test, we assume that the vestibular system is working because we do it on people with a normal functioning vestibular system. So we immediately know that the proprioception in case of swaying is not working correctly, and that there's a defect in the dorsal column system pathway. Moving on, why is it that when you touch someone, they would feel the exact area of where the skin was touched? However, they wouldn't feel it in the surrounding areas, even if the skin in the surrounding area is being moved or stimulated. This is because of something called lateral inhibition. So in the thalamus, we have something called relay nucleus, which has interneurons which can either be excitatory or inhibitory. And personally, I found the way this topic is presented in the slides a bit confusing, so I looked for other pictures to help explain it. Just know that this rule of thumb is that every presynaptic neuron would try to inhibit the other presynaptic neuron from moving on with their signals. Okay, so here we have the stimulus. We have the stimulus which would stimulate, let's say, this neuron, which is the main area where the stimulus is felt. The surrounding area also feels the stimulus, so the other neurons over here are stimulated. However, it is weaker, and the signal is not as strong as the main area. So it travels, and in the relay nucleus, we'd have interneurons from this neuron trying to inhibit the other presynaptic neurons. And even the other presynaptic neuron would try to inhibit the main one, the strong one. However, the strong one at the end would win and would advance with the signal. And the signal from the other neurons would not be able to advance. And this way we'd be able to tell the exact neuron which the stimulus was detected. This is known as lateral inhibition, and it happens in the thalamus and the relay nucleus, and at the end the neuron with the strongest signal wins. The purpose of this is to precisely localize the area of the stimulus and define the boundaries. So is it sharp or is it round or whatever? And this is for fine touch, because in crude touch, the boundaries of the stimulus aren't as clear. So lateral inhibition is for fine touch. Moving on to the next topic of receptive field. So receptive fields are areas in the body where, when stimulated, they only activate one sensory neuron each. So a receptive field can have hundreds of sensory receptors. However, all of these hundreds would only be supplied by one neuron. So any area stimulated over here would only be felt as one. And they come in different sizes depending on their location in the body. So in the back, you'd have the more larger receptive fields, and in areas like the fingertips, you'd have smaller field sizes. So in the back, because of that, in the back you'd have a lesser density because less neurons are in a specific area, and the opposite for the fingertips. So you'd have a greater density because you'd have more receptors crowded in a small area. So a receptive field acts as one unit. So as I said, if you touch anywhere, it would still stimulate the same neuron, meaning the brain wouldn't be able to tell that, okay, this stimulus is in this location or in this location or in this location. It would feel the whole area being stimulated, and it wouldn't even know the number of stimuluses. So all of these 4 stimuluses would be felt as one stimulus. So this is the concept of two-point discrimination. So for areas with larger receptive fields, at a certain distance, for example, let's say at 1 centimeter, if you have 2 stimuluses at 1 centimeter, they would still be felt as one touch because they're on the same receptive field. Even though there are 2 stimuluses, it's only one because it's one receptive field. However, in areas where there are smaller receptive fields at the same distance, let's say, and there are 2 stimuluses, they would be felt as 2 touches because they are at different receptive fields. So areas with smaller receptive fields like the tongue and the fingertips need a smaller distance to be able, a minimal of a small distance to be able to identify that there are 2 stimuluses instead of 1. However, for the areas with larger receptive fields, the minimum distance needed to identify that there are 2 stimuluses is actually longer. So to summarize, for the back of the torso, you have a bigger field size and a smaller density, and the 2-point discrimination range is actually larger. For the fingertips, you'd have a smaller field size, a larger density, and a smaller 2-point discrimination range. If you remember from foundation, we took something called temporal summation and something else called spatial summation. And the main difference is that temporal summation means in time. So in case a stimulus strength increases, the neuron firing rate increases in time. So the frequency of which a single neuron fires increases. However, for the spatial summation, spatial meaning in space, the number of the active receptors increases. So in compared to this over here, the space of the activated neurons increases. So in conclusion, a greater stimulus would generate a stronger response. And at the individual receptor level, this would be seen by more sodium channels opening. And at the receptor field level, this happens by temporal summation and spatial summation. So peripheral neuropathies are any conditions that affect the nerves. They can either be because of fractures which compress and damage a nerve, or they can be a side effect of certain drugs, or can be caused by conditions like rheumatoid arthritis, or by infections, or even by vitamin deficiencies, mainly vitamin B12 deficiency, because vitamin B12 is very important for the health of nerves. Now, I know I said I won't be explaining experiments, but I'll just explain the main concept of this experiment. Is that your brain does not care about the source where the information is coming from, and it only cares about the information it receives. So in theory, we would be able to replace our lost sensation by giving the brain ready information without depending on the same source of that information. So we have here, for example, a theory or an experiment of a blind woman, and we try to send the signals to the brain from the tongue. So we send through the glasses, we send the signals to the tongue, and we try to the tongue would try to send the signal to the brain, and then the woman would be able to see instead of using her eyes. And this is it for this lecture. I hope you found this video helpful.